Hello, Science 10, and welcome to the video on transports across membranes. We have learned about what a cell membrane is. Now we're going to discuss the different things that help us transport over a cell membrane, so like nutrients and ions. Today, your goal is to be able to describe and diagram diffusion, describe and diagram osmosis, explain the difference between them, explain what a concentration gradient is, and explain what active transport is. So we're going to start in the world right now of cell membranes and talking about semi-permeable membranes. So this is from yesterday. We learned that cell membranes only allow certain things to cross them. We need to figure out why does this happen and how does this happen. So it's all about this idea of high to low concentrations. And you may remember this when we talked about it in Science 9, or you've heard about it before in another grade, but essentially all molecules like to move from areas of high concentration to an area of low concentration. So chemicals are transported because of their chemical concentrations. So their concentration gradients. What a concentration gradient means is it's the difference in areas that you're looking at. So for example, in the pictures below, I have a concentration gradient in this picture because I have a massive amount of molecules on one side and none on the other. So this is called a concentration gradient. Cells are going to move from areas of high concentration to areas of low concentration. But eventually we know that cells will end up reaching something called homeostasis or balance. And that is when you see this. This is a no concentration gradient, meaning that they're equally distributed. For cells in the world of moving nutrients in and waste out, that is bad. You do not want to lose your concentration gradient if it is the reason you are able to move nutrients in, because then you would never eat. So concentration gradients are really important. So let's think about it in the world of humans and how we get on buses. If you think about how we load a bus, we all get in at the very front end of the bus. Yes, one by one, but essentially we start off pretty close, all of us, to the front of the bus. And eventually we move our way to the back. Think of the front of the bus where everyone's trying to pile onto it as the area of high concentration. And as we move to the back of the bus, this becomes our area of low concentration. Particles move the same way, but we don't always refer to it as high and low concentration. There's actually different terminology that we use in bio that helps us explain a concentration gradient. So if I see something is going up a concentration gradient, it means it's not favored. It requires energy. Think of climbing stairs. It takes way more energy to climb stairs than it does to quickly run down them, okay? So when you are moving up a concentration gradient, it's harder. So you need a little extra energy to do it. Sometimes we call this against a concentration gradient. So meaning it's going against, you're fighting to get across. Okay. So that's some of the terminology you may hear. Down a concentration gradient is a good thing. It's meaning it's moving from high to low. I'm going down my stairs. I'm going down to the bottom where there's nothing down there. Okay, so down a concentration gradient is really good because we don't need energy to do it. So within this lesson, we're going to talk about a couple different types of passive transport. Passive transport means no energy needed. You need to put that down because it's really important. No energy needed. Okay, so passive transport is in the world of diffusion, facilitative diffusion, and osmosis. And we're going to relate it to Brownian motion and again, keep talking about this idea of concentration gradients. So passive transport is one way cells move different types of molecules. The other way cells move different types of molecules is through active transport, in this idea of endo and exocytosis or using energy in channels like you see in the picture beside. It's moving of it from an area of low concentration to an area of high concentration. So we would say that's going against its concentration or up. So it's going to require energy. But before we do that, let's just stop again and talk about the cell membrane. The cell membrane's job is to distinguish between molecules, and it has to decide based on size, shape, and its charge. Again, how do we do this? Okay, well, essentially, we're going to say the smaller the size, the more likely it is to pass through with diffusion. The shape, the right shape will pass through a channel. If it's charged properly, okay, it'll be able to pass through it'll pass through the phospholipid bilayer, no problem. But again, it's the cell membrane's job to decide what is allowed in and what is not. and essentially out too. So let's connect this to Brownian motion. This is a concept that you kind of learned in grade 8 when you talked about particle theory. And particle theory states that all particles are always in constant and random motion. We know if we heat something up, it moves faster. If we cool it down, it goes slower. We know that solids, the movement is very small because they're compacted together. We know that in air, its movements are everywhere. So we know that they're always in motion. But what we also know is that they're constantly bumping into each other or bumping into the things around them. And what this does is it causes them to change position. So if you're looking at the picture here, the yellow particle is randomly moving around because it's being bumped by the water or the gas particles that are surrounding it. And as it's doing it, it's being pushed and shoved in different directions. So let's take this idea of Brownian motion, the random movement, and apply it to particles with diffusion. If you think about particles being stuck together and then being released, they're going to move from areas of high to low because they're going to bump into each other, change directions, and be pushed into places where there's less particles. So when we talk about that, we say a particle moves from an area of high concentration to low. As soon as there is particles everywhere, we call that equilibrium. So if you see what's happening in the picture here, those red particles are being struck by an unknown force. And when they are, they scatter because what happens is they want to move away from each other to find a space that they're comfortable in. So this results in what we would call diffusion, movement from high concentration to low concentration. So diffusion is one of our passive transports. And what it is it requires no energy at all. So particles move down their concentration gradient. Remember, down means from high to low. And really tiny small molecules are actually able to just slip through the phospholipid bilayer along their concentration gradient, so going down their concentration gradient. Now, the phospholipid bilayer allows them to pass through because remember, size matters. The smaller the molecule, the more likely it is to get in. If you're having a hard time picturing diffusion, kind of think of diffusion like a fart, okay? When someone farts in a room somewhere, okay, and it's not even near you, eventually you're going to smell it. Why? Because the particles are trying to move to an area where there's less of them. So it's really concentrated by where the fart is, but eventually those particles are going to spread out across the room, and you're eventually going to smell it because they moved from an area of high concentration to low, and it's going to be more near you where you weren't near the fart. Now, diffusion, high to low. Let's talk osmosis. Osmosis is also a passive transport, but it is specifically for the diffusion of water across a membrane. That is osmosis, okay? It has high to low concentrations, but it's only focusing on water. So we're going to use 3 terms when it comes to concentrations and osmosis: isotonic, hypertonic, and hypotonic. So in your notes, let's talk about these types of solutions. So the lower concentration of solute, so there's a lot more water molecules, so water flows out, is known as hypotonic. It's less concentrated, okay? Hypertonic is a high concentration of solutes but less water. And so what ends up happening is water moves towards it and flows into the situation. So then that way you can dilute those concentrations of solutes. So hypertonic means more concentrated. Hypotonic means less concentrated. So kind of think of it like this: a hyper kid with high sugar concentration. So hyper has high solute. So hyper, high sugar. The only way to calm them down is to give them lots of water. So bring water into their system. All right. And this is because they have too many solutes flowing around. So hyper and hypo are going to be words we work with a lot. If we are at a solution that has equal amounts of concentration on either side of a membrane, okay, then we call this isotonic. Hypertonic and hypotonic in the world of osmosis happen with membranes. So there has to be some sort of membrane, and one side has too much solute and the other doesn't have enough. And then we're going to have movement of water molecules based on that. So if you look at this cell right here, inside of the cell to outside of the cell, we're going to describe the situation using hypo and hyper. So outside the cell right here, okay, is hypotonic. Okay, a lot less solutes. So water is going to actually move this way. Okay? That's the way water's going to move. So when you move the water, okay, what ends up happening sometimes is cells get really full, like you would too if you drink too much water. And if they're not careful, they could burst. Okay, so cells have to be really smart about how much water they move in and out. So this one is hypo, meaning that it has way less concentration of solutes. That's the little things floating around in the black abyss. But the other side has way more. So it is known as hyper. And what we're trying to do is we're trying to clear out some of these solutes by bringing in water into the situation. So water is going to flow over into this side and fill it up. And what this does, again, like I said, ladies and gentlemen, is it just essentially allows for water to move down a concentration gradient from high concentration, lots of water, to low concentration, not a lot of water. And that is the world of osmosis. So when we talk about osmosis and diffusion, that happens with just passing through the phospholipid bilayer or a cell membrane. But sometimes that's not possible because sometimes there are things that are too big to fit through the phospholipid bilayer. So what ends up happening instead is we have to use some sort of assistance to pass you through, and we call these transport proteins. So if you look here, here's my phospholipid bilayer. These little molecules are able to just sneak through into the inside of the cell, okay? These ones are not. They're stuck because they're too big. They can't pass through that lipid bilayer. So instead, they find a channel, kind of like a tunnel, and they just go through the tunnel, and then they can come out the other side. It takes no effort whatsoever because it's still using a concentration gradient of high to low. So that leads us into active transport. And active transport now is all about energy, okay? We're going to do the practice pictures of osmosis and diffusion examples in a tiny bit. But for now, let's just talk active transport. So active transport is, in essence, what you need is energy. And the reason why you need energy is because you're going against your concentration gradient. You're moving up it, so you're climbing up the stairs. This also requires a membrane channel, but it also requires energy. So what you're seeing here is this ATP is energy. And what it's going to do is it's going to activate this channel to allow glucose to move inside the cell against its concentration gradient or up its concentration gradient. And that is because there was more glucose on the inside than there was the outside. So normally it should move from high to low, but it's not. Okay, so instead we activate a channel with energy, and then that energy carries the carrier protein. It carries it from low concentration to high. This leads us into the world of how do we get things in and out of the cell with active transport. And there's two types. There's endocytosis and exocytosis. I love both these words because I think they are so fun. But essentially, things are moved by endocytosis by creating a pocket around the material, folding up, and it becomes a vesicle. And then the cell can either eat it or drink it. So what happens is the plasma membrane or the cell membrane will actually steal and pick up large particles and essentially swallow it into the cell. When they do that, we call that eating, and that's called phagocytosis. If they're taking like a liquid in or something like drinking, we call that penocytosis. So a cell can use energy to bring in things in vesicles because they're too large to pass through the membrane or even a channel. The channel's too small. So we use this process to bring them inside the cell. When a cell eats or a large particle or drinks a large particle, like I said, it's taking in those two words, phagocytosis and penocytosis. I won't ask you, but you do need to know that they fall under the world of endocytosis. And endo means bringing in. Exocytosis, on the other hand, means to exit, to get rid of. So exocytosis means when you form a pocket around the material and then you take it to the cell membrane and let it go. Okay. Neurotransmitters, things that send signals in your brain, That's how they get passed between two different neurons, is they are sent into a space through exocytosis in which they diffuse over to the next neuron and attach. So exocytosis is used to get rid of things. Sometimes it's for positive things, or sometimes it's waste. Okay, never mind. Can't watch that. So this is a recap of both passive and active, and I'm going to link it into our video here. And I would like you to watch it to get a little bit more information. We are going to talk more about active and passive when we talk and do a little bit of an activity with osmosis and diffusion. If you have any questions, please let me know.